Hello, thank you for attending this webinar co-organized with our UK distributor, Caltag Net System, and set up by Selenis. Briefly, I will present you the company profile. So our goal is to accelerate the drug development by early selection of effective and targeted molecules in an optimization context of preclinical studies. For this, Selenis is developing innovative scaffold or matrix for culturing cells in 3D in a microenvironment that is more representative of the microenvironment found in vivo, and thus providing more predictive tools for any screening. Our three products are biomimesis hepatocytes, and biomimesis adipocyte, which I will present today. So what is biomimesis adipocyte? It's a new generation of scaffold for 3D cell culture. So our functionalized scaffold for 3D cell culture of mature adipocyte is made of hyaluronic acid, cross-linked with collagen 1 and 6, which are major components of the adipocyte extracellular matrix, where it plays an important role in connective tissue, also by providing growth factor, signaling between cells. So the hyaluronic acid is grafted with the RGDase peptides, which is the cell-binding domain of the extracellular matrix protein fibronectin. It is so cross-linked with both collagen 1 and 6, and the resulting scaffold is the biomimesis adipocyte, which you can see here on the scanning electron microscopy picture. We have the hyaluronic acid sheet, and here you can see the collagen chains, which we have artificially colored to make a better observation. The physical-chemical feature of this scaffold is very good porosity, ranging from 70 to 170 micrometers. The Young's modulus is about 0.50 kilopascal, which is very soft for this tissue, and it has a high swelling ratio because it is highly hydrophilic. So, as I told you that the biomimesis adipocyte is recapitulating the in vivo structure of the adipose tissue. You can see on this same picture a decellularized adipose tissue with a lot of fibers made especially of collagen and also hyaluronic acid. You can see the scale bar here, and here is our scaffold shown by the same observation. You can see the very high similarity in the structure of both our scaffold and the native decellularized adipose tissue. So we have a matrix that is really representative of the microenvironment found in adipose tissue. We can summarize three key features of our scaffold. First, it is natural and physiologic. So hyaluronic acid is a major component of the matrix, extracellular matrix, and collagen 1 and 6 are forming a fibrous network. That is very important, that is known in the literature to be very important for the adipocyte function and differentiation. So it will mimic the 3D cells microenvironment in terms of cell morphology, cell-cell interaction, cell matrix interaction, and protein and gene expression. So you can see here a bright field picture of differentiating human white pre-adipocyte cells in the scaffold, forming grafts full of lipids. I will show you more biological results in the presentation. The second key feature of our scaffold is that it is easy to use and is providing in a ready-to-use format. Here is a small video showing you how to use our scaffold. So how to use biomimesis? The scaffold is dehydrated and is provided in standard 96 well plates. You have no handling prior to use, just seed your cells directly onto the hydrogel. you place your cells into the incubator as usual. So after near 48 hours, cells have migrated into biomimesis. You can easily renew the medium by simply pipetting the old medium and refresh with new one. And then you can follow your cell growth directly with a standard microscope, because scaffold is translucent. You can just see here some hyaluronic acid chains and just set the focus to see your cells, which are growing in 3D. So you have to focus with the microscope to observe cells. Here it is. As you see, it is very easy to use. The third key feature of our scaffold, and maybe one of the most important, is that it is fully compatible with all standard analytical technologies. As you saw in the video, it is transparent, so you can perform any microscopic observation, either in bright field observation or fluorescence staining and observation. You can stain cells directly in the scaffold without retrieving cells before staining them. You can also use any plate reader for optical density measurement, fluorescence or luminescence measurement. The scaffold isn't generating any background, so you can use any detection technique you want. As you saw on the same picture, it is highly porous. So for any biomolecule extraction, nucleic acid, protein, or any quantitation of protein of interest like ELISA technique, you can perform all this procedure and extraction by adding your lysis buffer directly on cells growing inside the hydrogel. You don't need to retrieve cells before performing your extraction. Simply add your lysis buffer, follow your standard protocol, and retrieve your molecules. If you want to perform experimentation like flow cytometry, it is still possible with the hydrogel because it is biodegradable. We have a ready-to-use protocol for an enzymatic digestion, and you can retrieve cells with around 90% of cell viability. You can perform any analysis you want by seeding cells again or performing flow cytometry after any fixation or staining for the study of cells population. The scaffold is soft, but it can be embedded in paraffin, for example, to perform usual histology observation. You can see here the hematoxylin eosin staining of the scaffold after sectioning in microtome for histological study. You can stain cell or perform any immunocytochemical studies with cells growing inside the scaffold. So you don't have any technical limitation by using this scaffold. I will show you a few proof of concept that we obtain with culturing adipocyte in 3D. So here is the culture protocol that we used for the result I will show you. So just seed cells, and as usual, we perform seeding. We are working in 3D, so we need a few more cells than in 2D culture. So we saw 50,000 cells per hydrogel, then perform a growth period for 48 hours, then perform the differentiation of cells by using differentiation medium for 3 days, and then we have the nutrition phase with nutrition medium. That's where cells will accumulate triglyceride and become fully differentiated and mature adipocytes. So it is really a usual protocol. So you don't need to change your protocol to culture adipocytes. It's really a standard one that is already used in 2D for many labs. So when cells are grown in our scaffold, the first thing that we can see that they're acquiring an in vivo-like morphology. You can see here an in vivo scanning electron microscopy picture of an adipose tissue from human. You can see the very round and globular aspect of cells that is impossible to obtain in 2D. But when you are culturing cells in biomimesis adipocyte, either for the murine pre-adipocyte or human pre-adipocyte, you get a morphology very close to the one you can see on human tissue. By cells are forming grapes, multicellular aggregates, very round, and you can see here around them the scaffold. So cells are really growing, embedded in scaffold in interaction with the extracellular matrix provided by biomimesis adipocytes. So we really have an in vivo organization like in the adipose tissue that we obtain by culturing cells in biomimesis adipocytes. So cells are acquiring an in vivo-like morphology, and you can see here the round shape of multicellular aggregates growing in biomimesis adipocyte. This one is nearly 100 micrometers in diameter after 3 weeks of culture. So we monitor the spheroid diameters during the culture, and during the 4 week of culture, either for the human or the murine adipocyte, we have a continuous growth inside the scaffold, so attesting that cells have enough space to grow, because adipocytes are known to be quite big cells. So the highly porous structure of biomimesis allows cells to grow for more than 4 weeks. You can also perform culture for 6 or 8 weeks if you want. So we have a continuous growth with healthy multicellular aggregates. Of course, we perform live dead staining, and results attested that all cells are alive during this period. So the pre-adipocyte needs to be differentiated into fully mature adipocytes. So first, we monitor the expression of very well-known differentiation genes that are considered to be early marker of the adipogenesis processes. So CIDP alpha or PPAR gamma 2 and the fatty acid binding peptide 4 were used as the marker for early differentiation, and compare between the 2D classic plastic dish culture condition and biomimesis adipocyte. So for 4 weeks of culture, and you can clearly see on this RT-PCR data that the expression is really higher and occurs earlier in adipocytes grown in biomimesis adipocyte compared to the 2D condition. So here is a quantification of the FABP4 genes normalized with the housekeeping gene RNA polymerase 2, of course. And you can see that we have a statistically significant higher expression of this marker of differentiation. So we have an increased induction of the early differentiation gene when cells are grown in biomimesis adipocytes. So one of the main functions of adipocyte is the lipogenesis activity, so the ability to synthesize lipid and to store them. So here is a picture of fluorescently stained lipid with the adipored reagent. You can see here on this bright field picture of the 2D grown adipocytes that first we have a very fibroblast-like shape, very tight cells, and only a few ones are still positive for the lipid staining compared with the high density culture here. But when cells are grown in biomimesis adipocyte, nearly all the cells, all the multicellular aggregates that you can see on this bright field picture are positive for the lipid staining. So attesting a very more homogeneous population of adipocyte differentiating in vitro compared to the 2D condition where less than half of the cells are positive for the lipid staining. We have really high density of positive staining of cells positive for the lipid staining in 3D. Sorry. So behind this microscopic observation, we perform quantification of the differentiation process by plate reading of the staining of cell with the adipored reagent and normalize with the DNA amount, because in 3D cells often show less division compared to the 2D condition. The proliferation rate is quite lower in 3D compared to 2D, so the data has normalized with the DNA amount to make accurate comparison, of course. So you can clearly see during all the cell culture for 4 weeks in the mouse adipocyte and the human adipocyte that we have a statistically significant higher amount of lipid in cells growing in 3D. So when cells are growing in biomimesis adipocyte, they display a really higher lipid accumulation compared to the 2D condition, more than 20 to 30 times higher in 3D than compared to the 2D condition. So to go deeper into the differentiation and maturation processes, we monitor the perilipin expression. Perilipin is a lipid droplet associated protein, which is expressed when the lipid droplets are growing inside, and this protein is stabilizing the lipid droplets. So you can clearly see here on this section performed by laser scanning confocal microscopy, you have two nuclei in red, and the perilipin is stained in green. So you can clearly see the droplets that are surrounded by the perilipin protein, attesting that we have a full maturation of the lipid vesicle in the human right pre-adipocyte grown in biomimesis adipocyte. And also, this kind of picture is obtained by directly staining cells still in culture in the scaffold, as I told you previously. You don't need to retrieve cells to perform this kind of staining. Just add your antibody. Okay, so there are again a few genes that are well known in the literature to be marker of the maturation of the functional adipocyte, which are called the late markers in adipocyte differentiation, and GLUT4 is one of them. So here are the RT-PCR data obtained with cells growing in our scaffold, the biomimesis adipocyte, compared to the 2D condition. Here is a typical RT-PCR obtained with such cultures, and the quantitation with the scanning and logiciel acquisition. And then you can see that, again, for this maturation gene, GLUT4, we have a high expression in the adipocyte grown in our scaffold compared to the 2D condition, that attesting that cells growing in our scaffold are more differentiated and are more adipocyte compared to the 2D condition. And here is a picture of lipid stained cells, and for the late maturation of cells after more than 4 weeks of culture, we can get some unilocular adipocytes, which confirms the maturation of these cells in 3D compared to the 2D condition, which is it's impossible to get unilocular adipocyte in 2D condition. So now we have some fully functional adipocytes. So what can we do with them? We can use them as a lipogenesis model to study the regulation of these processes. So we compare the inhibition of lipogenesis with or without the retinoic acid, which is a well-known inhibitor of lipogenesis. So here is in 2D condition without retinoic acid, and the clear gray is with 1 micromolar of retinoic acid, and you can see that we have a strong inhibition of the lipogenesis process in 2D with just 1 micromolar of retinoic acid, despite in 3D in biomimesis adipocyte, when cells are exposed to retinoic acid, we attain only a very weak inhibition with 1 micromolar of retinoic acid. For 1 micromolar, we get 65% inhibition in 2D, but only 30% inhibition in 3D. And to get a high inhibition of lipogenesis, we got to increase the concentration of retinoic acid or up to 5 or 10 micromolar. So for this kind of molecule, it may be a penetration issue, but it's also the way that cell will perform in vivo. So biomimesis adipocyte is really a robust model for studying lipogenesis assay and dose effect on this process, unlike the 2D culture, whereas only very weak dose of inhibitor lead to a very strong decrease in the lipogenesis. So the 3D is really better for studying this kind of process. And last, a fully mature adipocyte should be able to mobilize triglycerides, a process that is called lipolysis. So a lot of drug screening campaigns are about the lipolysis. So we monitor the lipolysis by measuring the glycerol release during this process. So here we have the control untreated, and then the isoprenaline treatment during 90 minutes. The isoprenaline, that is a well-known inducer of the process of lipolysis. So after 2 days in culture, so we study the release of glycerol stimulated by the isoprenaline on murine adipocyte grown in our scaffold, and we have a dose effect response with the isoprenaline and also with the lipolysis inducer caffeine, which is a very well-known inducer. We are working with quite high concentration in 3D, but we have a 50 to 60% increase in released glycerol after this inducer treatment, thus confirming that cells grown in biomimesis adipocyte constitute a very relevant model for lipogenesis and dose effect analysis. Okay, so here we are. And so biomimesis adipocyte is provided in a low attachment plate format. It can be either full, containing 96 hydrogels per plate, or you can get half plates with 48 hydrogels or 24 hydrogels provided in a low attachment plate. Our product is also available in black plate to perform fluorescence measurement directly on the black plate. And we have a quality process that ensure you to get quality control batch and barcoded plates for this. So to summarize the result I've presented to you today, so the cells growth in biomimesis adipocytes acquired an in vivo-like organization with spheroid-forming multicellular aggregates. The cells also have a significantly higher lipogenesis activity, which is up to 25-fold higher than in 2D culture. The early differentiation gene expression is also higher in our scaffold, and the complete maturation of adipocyte is achieved compared to the 2D culture condition. And the lipid accumulation of the adipocyte in culture can be regulated either as a lipogenesis phase or for the lipolysis process also. Okay, so thank again to our distributor, Caltag Next System, and for the UK territory. If you have any questions or any order, please contact Caltag, and here you have the email address. And if you're not in the UK, you can easily contact us by sending an email to this address at contact@altselenis.com. Thank you. Now I have some time if you have any question. Okay, so first, where did you purchase the cells? So to allow our customer to get the same result as us, so we worked with commercial products. So we bought cells to process providers, and we also use a fully commercial medium. And if anyone is interested to get the full composition of the growth, differentiation, and the maturation medium, you can send us an email and we provide you the full composition of the medium. There is a question in Spanish, but I'm very sorry. Can you please ask your question in English? Oh, okay. Would you address? So it is asked me to show the lipogenesis process again, so no problem. Okay. Okay. So the lipogenesis activity, where we first monitor this activity by staining lipid with fluorescent dye and compare our 3D grown adipocyte to the 2D grown one. So you can see here on this confluent culture of adipocyte in Bradfield that first we have a fibroblast-like shape after 2 days of culture. And when cells are stained for the lipid staining, you can see that we have only a very few cells that are positive for lipid staining after 2 weeks of maturation, contrary to the 3D condition of cells grown in biomimesis adipocytes that nearly all of the multicellular aggregates you can see here are positive for lipid staining, and it's the same culture times that culture were really performed in parallel. So with the same vial of cells that was split in 2D or in biomimesis adipocytes to allow direct comparison. And we quantified this staining by quantifying the fluorescence with fluorescent plate reader and normalize the data with the DNA amount. As I told previously, in 2D cells are often proliferating faster than in 3D. So we quantify the DNA amount of each condition to normalize the data for lipid quantification. And either for the murine adipocyte and the human adipocyte, you can clearly see that we have a very high amount of lipid in 3D compared to the 2D condition, which is, of course, statistically significant in both murine and human adipocytes. So we have a really higher lipid accumulation in biomimesis adipocyte compared to the 2D condition. So hope it answered to the question. Don't hesitate if you want me to go back to any slides or any results. I will be happy to present it to you again. So why to choose the RP2 housekeeping gene? The GAPDH is often used for housekeeping gene, but we thought that this gene, this gene expression is not consistent between 2D and 3D condition. So that's why we chose to work with the RNA polymerase II for the housekeeping gene, because its expression is really stable during time and culture condition. How to stain it with 3D situation? Okay. The staining, I will show you the staining. So I just searching for the pyridipine staining. So the immunofluorescence staining is really performed as usual. So here is an example of what you can perform for the immunofluorescence staining. So we choose a pyridipine because it's a lipid droplet associated protein, but cells are, for all the procedure, all the protocol, cells are still in the scaffold. That's really easy to perform. Just fix cells with any fixator like PA, like the paraformaldehyde, permeabilization step, saturation step, antibody incubation, washes, and microscopic observation and acquisition. All steps are performed with cells still in culture in the scaffold, which is really easy because you can handle it with fine scissor and put on your microscopic glass if you want to perform microscopic acquisition, like on this one with the laser scanning confocal microscopy. So the protocol is roughly the same than in 2D, no difference. English too, let's speak. Okay, so there is a question in Spanish, so sorry. Maybe is it okay that you wanted to talk about the histology? And maybe it's the only word I understand in Spanish, histology. There is a question about differences in between male and female, or also black people. So really like I cannot answer this because we worked with human white pre-adipocyte. As I told you, we purchase cells on the commercial provider of cells. So I think if you want to know more about the cells available, you can contact directly Promocell, which is a provider of cells. We worked with the IMC, with standard IMC people, because people working on obesity or any other disease, you can ask to purchase cells from donor with a particular disease, but we work with a healthy donor, and I don't know if it's possible to get some cells from Black people. Should be available, but we work with human white pre-adipocyte from male donor without any disease. Histology. So there is a question for the histology. So as I told previously, yeah, the hydrogel is soft, but you can handle it with scissor and you can embed it in a paraffin. The time required is advised to be overnight to have a fully embedded hydrogel, and you can perform sectioning with some slice around 5 micrometers. And yes, you can perform any histological study. Is it possible to cultivate other cell types? Yes. The scaffold is designed to be optimal in terms of microenvironment for adipocytes, but for any cells for which the collagen 1 or the collagen 6 may be of interest, should be cultured in our scaffold. It is fully biocompatible, and I know that we have a customer who is studying the breast cancer cell and adipocyte interaction, and she is working with biomimesis adipocyte to grow both cancer cell line and adipocytes together to study the interaction. So this kind of scaffold also is really it's really easy to perform co-cultures also. Okay, so is there any question? I have another question in Spanish. I'm sorry, I'm not sure to understand. Is it still need SDM? Can you please precise me what is SDN, please? Is it growth factor or not? Okay, for excuse me, for the person attending who is asking question in Spanish, we will answer to you just a bit later, so we have time to translate accurately your question and provide you an accurate answer also. So we will answer to you later, please. Standard differentiation medium. Yes, it may depend on cells, and we saw some cells able to display small lipid droplets without differentiation medium, but we follow the standard culture procedure with first a growth medium, differentiation medium, and maturation medium. So yes, we use standard differentiation medium and strictly compare the 2D condition to the 3D scaffold condition with the same culture protocol and the same medium. So yes, you need standard differentiation medium. If you want us to communicate to you the exact composition, we can provide you the composition. Okay, don't hesitate if you have any question. I still have a few minutes to answer. Okay, thank you for attending this webinar. Don't hesitate to contact us for any information. Bye.